Inside Science.
Welcome to the floor of the Pacific Ocean. It's dark and it's cold, but buried in the oxygen-poor mud may be a crucial part of the story of how complex life evolved on Earth. When Japanese researchers carefully cultured samples from this sediment, they found a novel kind of archaea, ancient single-celled organisms. It's thought that more than 2 billion years ago, an archaea like this somehow evolved into the first complex eukaryotic cell that would go on to make up modern plants, animals, and fungi. Now, new research claims to show us how. In the early seas, archaeans like this had none of the complicated internal machinery that modern eukaryotes have. So when they produced waste hydrogen, they needed nearby bacteria to break it down. In return, the bacteria released nutrients. So the archaea might have enmeshed those bacteria with tentacle-like processes to increase the efficiency of transfer. But roughly 2.7 billion years ago, toxic oxygen began to build up in the atmosphere, so the archaea needed to ensnare a new companion to help. Now, the researchers suggest that this new relationship became so close that the constant fatty bubbles trafficking nutrients between archaea and bacteria eventually closed over them both and became the beginnings of the cell membrane. And voila, the eukaryotes were born. Now, the archaea in this study are distant relatives of our billions-year-old ancestors, so their biology is unlikely to be identical. But they do share some ancient DNA with eukaryotes, and the researchers are hopeful that they will offer an accurate window into the past. So much so that they've named the new cell Prometheo-Archaea, after the Greek Titan who brought fire to the ancient world. In fact, you can see what could be the descendants of those early nutrient bubbles in modern cells, and a new microscope technique lets you do so in unprecedented detail. It works by tagging specific structures with fluorescent molecules, then flash freezing the cells and imaging at very high resolution, slicing off layers one by one to get to the next. Now, I think this kind of work is beautiful in its own right, but understanding the micro world is incredibly important for understanding the rest of the world. And if you don't believe me, when this video is done, read this article on the Inside Science website about how viruses can affect the entire global climate. And speaking of, the biggest news this month was the Australian wildfires, and a recent rapid response review confirms that such fires have become more likely because of climate change. So we can look forward to double the amount of Amazonian wildfires by 2050, and in the US, researchers predict tens of thousands more violent crimes. Now, they assume that higher temperatures will lead to milder winters, so more people and more criminals will be outside, therefore the chance of the two meeting and crimes occurring will go up. So, yay. But let's finish on a positive note. Sarin gas. A US Army research team has developed a gene-editing virus that can neutralize its deadly effects. Injecting the virus into mice allowed their liver cells to release paraoxonase 1 into their bloodstream. Paraoxonase 1 is a chemical that safely scavenges a variety of nerve agents, including sarin, before they can do any harm. And the mice's immunity lasted for months. And not only could this concept potentially protect us against terror attacks, it might also help guard farmers from the toxic effects of various pesticides. And you can read more about that in this Inside Science article. But that's it from me. Let me just leave you with the sound of a 3,000-year-old mummy whose larynx has been CT scanned and recreated with a 3D printer. Ladies and gentlemen, I give you Egyptian priest Nesyamun. Goodbye.
Inside Science. If you enjoyed this edition, follow us on the web and social media. Powered by the American Institute of Physics and a coalition of underwriters.